Cholangiocarcinoma, malignancy of the bile duct, is increasing in incidence. Cholangiocarcinoma is now the leading cause of primary liver cell death, having overtaken hepatocellular carcinoma in the UK. There are intrahepatic, hilar, and extrahepatic forms. Intrahepatic cholangiocarcinoma incidence is soaring worldwide. It remains resistant to efforts to achieve long-term survival. Surgery is the only curative strategy with poor 5-year survival, and only 6% of patients are amenable to resection. Current chemo, radio, and photodynamic therapy is non-curative. New adjunctive strategies are urgently needed. Cholangiocarcinoma is a good example of a solid tumour and consists of malignant epithelial cells surrounded by macrophages, myofibroblasts, and extracellular matrix with which it interacts. This is known as the tumor-stromal microenvironment. In general tumour biology, there's recent evidence that tumour progression is promoted by the surrounding cellular niche. Little is known about the role of the microenvironment in cholangiocarcinoma. In solid organ tumours, these cells are thought to partly derive from the bone marrow, with about 40% of myofibroblasts from the mesenchymal compartment and 30% of macrophages from the hematopoietic compartment. And even some reports suggesting that malignant epithelial cells may be derived from bone marrow. An understanding of the origin of tumour cells is important in determining therapies, and currently there's experimental interest in developing drugs that target the tumour microenvironment. The role of bone marrow cells is not known in cholangiocarcinoma. The Notch signaling pathway consists of 4 receptors, 5 ligands, and allows cells that directly contact each other to communicate. It's essential in embryonic development, but in cancer, abnormal Notch signaling has either an oncogenic or tumor suppressant role depending on its tumor type. The role of Notch is not known in cholangiocarcinoma. Our group has identified that in the context of inflammation, a microenvironment exists around the regenerative stem cell niche in the liver, and that Notch signaling promotes biliary differentiation towards a normal cholangiocytic phenotype. The Notch ligands, jagged, that signal this are donated from the myofibroblasts. In view of this, our hypothesis is that intrahepatic cholangiocarcinoma is a disordered regenerative microenvironment driven by Notch signaling between stroma and malignant epithelial cells, and that the stromal microenvironment is in part derived from bone marrow. Our aims are to identify whether Notch signaling is upregulated in cholangio, to define the role of Notch signaling on disease progression in intrahepatic cholangiocarcinoma, to assess the potential of pharmacological Notch ablation with gamma secretase inhibitors, and to elucidate the origin of the cells contributing to the tumour microenvironment. Following surgical resection, I obtained samples of human cholangiocarcinoma and normal liver from the same patients. Using a gene array, we demonstrated that the Notch pathway is upregulated, specifically the Notch3 receptor, jagged 1 and 2 ligands, and also downstream effector targets. To confirm these findings, we performed qPCR to look at gene expression, which identified a similar pattern for the individual receptors, ligands, and downstream targets. We then looked at the protein expression of the Notch pathway in the human lesions and found minimal Notch receptor and ligand in normal human liver and biliary tissue. When we looked at the cancers themselves, we were expecting to see Notch ligands on the stroma and receptors confined to the epithelial cells. We found that, as we suspected, the ligand, jagged 1, was expressed exclusively on the stroma, specifically on the myofibroblasts. But interestingly, the Notch3 receptor was expressed on clonal subsets of the epithelial cells, suggesting a heterogeneity within the malignant bile ducts, and also on a subpopulation of myofibroblasts close to the bile ducts. And this is confirmed on dual staining for both Notch3 receptor and jagged 1 ligand. To investigate the formation of intrahepatic cholangiocarcinoma over time, I developed a model system in which rats fed thiacetamide, which is a hydrogen sulfide donor, reliably developed intrahepatic cholangiocarcinoma in a stepwise fashion over 26 weeks. The tumours had excellent histological similarity to human lesions and characteristic MRI and small animal ultrasound features. Rats were harvested at serial time points during tumour formation. Using a gene array, we identified over time from normal to inflamed, dysplastic, and then established malignant tissue that the Notch3 receptor, Notch ligands, and also downstream effectors were noticeably upregulated in comparison to other components of the Notch pathway. Progressive disarray of the biliary architecture occurs during TAA cholangiocarcinogenesis. In parallel with these histological changes, there's an anatomical clustering of jagged ligand in the fibrotic and subsequently cancer-associated fibroblasts, together with an upregulation of Notch3 expression on fibroblasts and then cholangiocyte subpopulations in malignancy. And the final histology in the rat matched that of the human lesions. By having demonstrated upregulated Notch signaling in both human and rat cholangiocarcinoma, we then looked in vitro at cholangiocarcinoma cell lines to assess chemosensitivity. We first showed that Notch receptors were expressed at a gene and protein level in our lines, and then using a transfected reporter construct, confirmed that the cell lines are responsive to pathway inhibition using DAPT, which is a gamma secretase inhibitor, and downregulates the Notch pathway. DAPT is a potential chemotherapeutic agent, and gamma secretase inhibitors are currently undergoing trials in other tumours. I also showed that DAPT inhibits cell proliferation in vitro compared to controls at 48 hours. Gamma secretase inhibition in vitro appears to augment chemosensitivity. Combining DAPT treatment with 5-FU or cisplatin resulted in a significant decrease in the chemotherapy dose required to inhibit proliferation. Inhibitory IC50s trend towards reduction. Importantly, specific inhibition of Notch3 using RNA interference also enhances 5-FU chemotherapeutic effects. Well, to investigate the origin of these cells within the tumour microenvironment, I developed a bone marrow transplant model where we irradiated normal rats and transplanted the bone marrow from transgenic green fluorescent protein rats. Rats recovered for 10 weeks with the formation of bone marrow chimeras with successful reconstitution of both the hematopoietic and mesenchymal compartments. These chimeras were then given TAA and allowed to develop cholangiocarcinoma in a progressive fashion. We were able to track what cells in the tumour microenvironment were green, and so therefore bone marrow derived. Bone marrow-derived cells are orange in the merged image on the left-hand side. They stain a combination of green for the fluorescent bone marrow and red for an antibody that has been used to specifically mark the cells of interest. We found that the overwhelming majority of hematopoietic cells, that is macrophages and neutrophils, were bone marrow-derived. Macrophages did not express the Notch ligands. In contrast, and unlike different groups studying other tumours, we found that mesenchymal cells, myofibroblasts, stained only red and not green, meaning they're locally derived and not migrating from the bone marrow. These are the cells that donate the Notch ligands in the tumours. Using two different staining markers for epithelial cells, we confirmed that the bone marrow-derived cells did not contribute to the malignant epithelium of intrahepatic cholangiocarcinoma, but that these arose from the local epithelium, and there was no relationship with Notch signalling. So in conclusion, JAGID1 Notch3 signaling is active in the tumour microenvironment of cholangiocarcinoma in human and rat. The pathway is upregulated through disease progression, but the cellular expression appears more complex than the regenerative niche in normal liver. Notch inhibition restricts proliferation of intrahepatic cholangiocarcinoma and appears to enhance chemosensitivity in vitro, and further work is ongoing with some in vivo models. Bone marrow-derived cells contribute to the hematopoietic compartment, but not the mesenchymal compartments or malignant epithelium of tumours, and they do not appear to contribute to Notch signalling. I'm very grateful to all of those who've been involved with work with me as well. I'll answer your questions. Thanks.